So we can move towards the fourth point, photosynthetic pigments. In this point, we'll see the four types of pigments in the chloroplast. The first one is chlorophyll, second one is carotenoids, third one is phycobilins, and the fourth one is anthocyanin. We'll see the first point. In this point, we'll see the types of chlorophyll. The chlorophyll 9 types was invented by the Aronoff and Alin. The first chlorophyll is chlorophyll-a, chlorophyll-b, chlorophyll-c, chlorophyll-d, and chlorophyll-e, and bacteriochlorophyll-a, bacteriochlorophyll-b, bacteriochlorophyll-c, bacteriochlorophyll-d, and bacteriochlorophyll-e, and last one is bacteriochlorophyll-g. List one this. And about chlorophyll, we'll see it is most important and it is the most abundant photosynthetic pigment. It is a green pigment. It is soluble in organic solvent and insoluble in water. Chlorophyll A and chlorophyll B are present in higher plants and green algae. Next, about chlorophyll A and chlorophyll B. Chlorophyll A is blue-green pigment and chlorophyll B is yellowish-green pigment. Distinguish between chlorophyll A and chlorophyll B. The empirical formula is, we'll see here empirical formula and the empirical formula of chlorophyll B. It is, the difference is 1 here 72 hydrogen and here 70 hydrogen, not oxygen, here hydrogen is. And on the third carbon of the chlorophyll A, the CH3 group means methyl group is present, and here aldehyde group is present on the third carbon. It is present in chlorophyll A is present in all photosynthetic organisms except photosynthetic bacteria, and chlorophyll B is present in higher plants, green algae, bryophytes, and all vascular plants. And chlorophyll A converts light energy into chemical energy, and chlorophyll B transfers absorbed light to chlorophyll A. And chlorophyll A is essential pigment or primary photosynthetic pigment, and chlorophyll B is accessory pigment. Go to the next. About some important information, general knowledge. Chlorophyll A and chlorophyll B both shows maximum absorption is blue, violet, and red region of visible spectrum. In algae, chlorophyll C, chlorophyll D, and chlorophyll E is present. Also, it contains chlorophyll A. And two terms which are important for entrances. The first one is bacteriochlorophyll, which is purple sulfur bacteria, which is chromatium chlorophyll, and bacteriopyridine, which is green sulfur bacteria, which is chlorobium chlorophyll. To the next, here the structure of let's see the structure of chlorophyll. This is the chlorophyll A and this is the chlorophyll B. You see the some list one difference of chlorophyll A and B. Here the methyl group is present and here the aldehyde group is present. This is chlorophyll A and this is chlorophyll B. At the middle the magnesium element is present. Magnesium is present. Next, carotenoids. These are the carotenoids. About carotenoids, it is fat soluble one. It is lipid compound and it is distributed in chloroplast and chromoplast. The functions of carotenoids: primarily it absorbs blue-violet region of visible spectrum, and it transfers light into the chlorophyll A. Thus, it functions as accessory pigment. It protects chlorophyll A from photooxidation. Two types of carotenoids. The first one is carotenes and the second one is xanthophylls. First one is orange the carotenes are orange in color, xanthophylls are yellow in color, and carotenes are unsaturated hydrocarbons. We'll see the empirical formula C40 and H56, and the xanthophylls are oxidized form of carotenes, also called carotenol, and in the empirical formula is C40, H56 and O2. The carotenes are insoluble in water and soluble in organic solvents, and the xanthophylls are insoluble in water and soluble in organic solvents. The carotenes. The beta carotene is the major carotene found in the plants, and the lutein is major xanthophyll found in the plants. The beta carotene and lutein is important one. As next, we'll see the beta carotene and the xanthophylls. Here see the OH group is present, and this is the xanthophyll. Phycobilins. About phycobilins. Phycobilins are the accessory pigments which are present only in cyanobacteria and red algae. Water soluble pigments. Phycobilins are the water soluble pigments, and the examples of the phycobilins are phycocyanins and phycoerythrins. Phycocyanins are blue in color and phycoerythrins are red in color. Erythra means RBC. We'll see in the class 12th circulation chapter RBC red in color. Then the name gives red, phycoerythrins, and cyanin means blue. Next, we'll see the anthocyanin. It is a purple in color. It is present only in higher plants, and in flowers it is non-photosynthetic. The phycocyanin in flowers it is non-photosynthetic. We'll go to the next, light reaction. We'll see about light reaction. Light reaction is also called as primary photochemical reaction, also called as Hill's reaction and Arnold cycle. Light reaction is also called Hill's reaction. The chloroplast produce oxygen from water in the presence of light, and light reaction is also called Arnon cycle. Arnon showed that the H plus ions released by the breakdown of water are used to reduce the coenzyme NADP to NADPH2. You see here photophosphorylation. You see the definition of photophosphorylation: the process in which energy rich ATP molecules are synthesized by using solar energy is called as photophosphorylation. The types of photophosphorylations are: first one is cyclic photophosphorylation and the second one is non-cyclic photophosphorylation. You see the first one is cyclic photophosphorylation. The definition of cyclic photophosphorylation is it is the type of photophosphorylation in which the expelled electron from the chlorophyll A is returned back to the same chlorophyll A following a cyclic path is called cyclic photophosphorylation. You see the diagrammatic representation of the cyclic photophosphorylation. Here it is the cyclic photophosphorylation. According to the definition, the electron from chlorophyll A come back towards the same chlorophyll A and following a cyclic pathway. Hence it is known as cyclic photophosphorylation. This is the photosystem 1. This is upper reaction center. These are the accessory pigments P700 chlorophyll A and photons of light. And here the photo excitation takes place and electrons goes into the first electron acceptor FRS, ferredoxin, reducing substrate, and then it goes into ferredoxin, and here the ATP molecule is formed and cytochrome B6 to cytochrome F, also one ATP is formed, and then it goes into plastocyanin, accepts electron, and it transfers de-energized electron into photosystem 1 for energized. Here we see the mechanism, the 5 steps. In the first one is absorption of light energy, second one is photoexcitation of PS1, third one is transfer of energized electron, fourth one is generation of ATP and transfer of de-energized electron. Here see the first step is absorption of light energy. First absorbs light energy, and the second step is here the photoexcitation takes place and the energized electron. Third one is transfer of energized electron, first towards FRS, then here, and the fourth step is generation of ATP. There are two ATP molecules are generated in the cyclic photophosphorylation. The first in between pyridoxine and cytochrome B6, and the second in between cytochrome B6 and cytochrome F. And the last step is the transfer of de-energized electron towards the PS1. Here the energy is lost for the formation of ATP, and de-energized electron goes back towards the PS1 for gaining energy. And go towards the non-cyclic photophosphorylation. Let's see the definition of non-cyclic photophosphorylation. It is the type of photophosphorylation in which the expelled electron from chlorophyll A is never returned back to the same chlorophyll A is called a non-cyclic photophosphorylation. It involves continuous flow of electrons, and the electrons never return back to the same chlorophyll. Hence it is called as non-cyclic photophosphorylation. And the diagrammatic representation of the non-cyclic photophosphorylation, see here. It is also called a Z scheme, name given by Hill and Bendel. Here two photosystems are comes, photosystem 1 and photosystem 2. And the first is the electrons goes here, and then here. So we'll see first mechanism and then explain. There are 8 steps in the mechanism. First one is photoexcitation of PS2, second one is transfer of energized electron, third one is generation of ATP, and the fourth one is transfer of de-energized electron, fifth one is photoexcitation of PS1, and the sixth one is transfer of energized electron, and the seventh one is generation of NADPS2, and the last one is photolysis of water. We'll see in the diagrammatic representation. First it absorbs the light energy, and then the photoexcitation of PS2 takes place. Here the reaction center is P618, and the energized electron goes into the coenzyme Q, then plastocinin, then cytochrome B6, then cytochrome F. Here the generation of ATP takes place, and then de-energized electron come back toward the plastocinin and then goes into the photosystem 1 for gaining energy. Here again the absorption of energy takes place, and the photoexcitation of PS1, and then energized electron goes into transfer of energized electron. First goes into FRS, then FD, pyridoxine, and then here NADP comes, and the 2 NADP here, and the 4 hydrogen takes place, and here the NADPH2 generated. 2 molecules of NADPH2 is generated. Here the photolysis of water takes place, the process of photolysis, and these are the electron acceptors. To the next, about photolysis of water, we'll see the photolysis of water. P680 acts as a strong oxidizing agent and brings about splitting of water molecule to release oxygen with the help of light energy. This light dependent splitting of water molecule is called photolysis of water. This is important one. Magnesium, calcium and chloride ions play an important role in photolysis of water. Here the reaction of photolysis of water, 4H2O gets converted into gives by in the presence of light and chlorophyll 4 electron molecules which goes into the PS2 and 4 hydrogen atoms which goes towards the 2 NADPH molecule and 2 water molecules and oxygen involved. And when 4 electrons are removed from the reaction center of PS2, it induces splitting of 4 water molecules into 4 hydrogen atoms and 4 OH. The hydroxyl ion thus formed donate electrons to the reaction center P680, and the 4OH radicals react amongst themselves to form 2 water molecules and oxygen get released. Here we'll see the difference between some cyclic photophosphorylation and non-cyclic photophosphorylation. Electron travels in cyclic manner, here non-cyclic manner, and energy rich molecule ATP is formed. Here both ATP and NADPH are formed. Photolysis of water doesn't take place in cyclic, and in non-cyclic photophosphorylation photolysis occurs. Cyclic photophosphorylation only PS1 is involved, and in non-cyclic photophosphorylation both PS1 and PS2 are involved. And then we'll see the significance of light reaction. The generation of assimilatory power. Second, it generates assimilatory power means ATP and NADPH2. And second one is it releases free molecular oxygen in the environment. Third one is photolysis of water takes place, and water acts as a donor of hydrogen proton for the reduction of NADP. Here in the non-cyclic photophosphorylation, the photolysis of water takes place. And the fourth one is NADPH2 acts as a hydrogen donor in the dark reaction, with the help of in which the CO2 gets reduced to carbohydrate. The NADPH2 is used in the dark reaction in Calvin cycle. And the fifth one is ATP acts as energy donor in the dark reaction. These are the significance